A very warm welcome, dear children. We will start with the next type of cell division, that is meiosis. In the previous class, we studied about mitosis, which takes place in the somatic cells, and it is also known as the equational division. So now we will study meiosis. Now, meiosis was discovered and described for the first time in an organism which is known as sea urchin, right? So for the first time, meiosis was discovered and described in sea urchin in its eggs, right? So in the eggs of sea urchin. Now this was for the first time described by a German biologist. He is Oscar Hertwig. So he was the German biologist who had studied meiosis. He discovered and described this for the first time, right? So in which organism? In the eggs of sea urchin. Then again this was discovered in around 1876, Oscar Hertwig. Later on, this was again described in 1883. So again in 1883 at the level of chromosomes, right? So this was described keeping in mind the chromosomes. So at the level of chromosomes, this was described by a Belgian zoologist. So he was known as Edward, slightly the spelling is different, Edward van Beneden, right? So he had described this, he had studied this, keeping in mind the chromosomes in the eggs of Ascaris, right? So Ascaris you've already studied. The significance of meiosis for reproduction and inheritance, right? So why is meiosis important? Why do we study, or what is its significance? So its significance mainly is that it plays a role in reproduction. Meiosis is related to reproduction and inheritance, right? So based on this, meiosis was described only in 1890 by a German biologist, August Weismann, W-E-I-S-C-M-E-M-A-N-N, August Weismann, right? So significance of meiosis is related to reproduction and inheritance, and this particular part was described in 1890 by August Weismann. That means August Weismann was the biologist who described the importance of significance in reproduction and inheritance. Now, he also noted down that 2 cell divisions were necessary. Then again, what is another important thing that takes place in meiosis is that there are 2 cell divisions, right? So 2 cell divisions take place in meiosis to transform 1 diploid cell into 4 haploid. So 1 diploid cell is transformed into 4 haploid cells, right? 4 haploid. This leads to the formation of 1, 2, 3 and 4, right? 4 haploid cells, right? If the number of chromosome has to be maintained, right? So what do we study is that in any species that we talk about, the number of chromosome is maintained in every generation, right? If that gets disturbed, then that leads to some kind of a defect. So if we talk about human beings, there are 23 pairs of chromosomes, right? So every organism, every human being will have 23 pair of, normal human being will have 23 pair of chromosomes, right? Now if we talk about the gametes have half the number of chromosomes. That means they have only 23 chromosomes. Here when we say 23 pair of chromosomes, it means 46 chromosomes, that is a diploid cell, and the gametes, that is the sperm and the ovum, will have 23 chromosomes. Now if this number has to be maintained, then he said that a diploid cell has to undergo 2 cell divisions, which will lead to the formation of 4 haploid cells, right? Now in 1911, an American geneticist named as Thomas Hunt Morgan, right, that is his name, Thomas Hunt Morgan, he observed crossover. We will study what is crossover in the fruit fly, right? What is the fruit fly scientific name? Drosophila melanogaster, right? He observed meiosis and provided the first genetic evidence that genes are transmitted on chromosomes. So it was the scientist who gave the evidence that genes are transmitted on chromosomes, right? So genes are transmitted on chromosomes. Now coming to the term meiosis, right? The word meiosis is derived from a word meio, M-E-I-O, which means less, and this term was coined by means this name was given by J.B. Farmer and J.E.S. Moore, right, in 1905. The cell which undergoes meiosis is known as meiocyte. So meiocyte is the cell that undergoes meiosis, right? So a cell has to divide to undergo meiosis, so the cell that divides is known as a meiocyte. Meiosis is also known as, there is another name for meiosis, this is also known as reduction division, right? So one cell, diploid cell, when it undergoes meiosis, it leads to the formation of 4 daughter cells, right? So reduction division, because it causes reduction in chromosome number. Now where does this occur? It occurs in the germ cells, right? Occurrence, right? So where does it occur? Where does this kind of cell division take place? It takes place in the germ cells. What are germ cells? That which give rise to gametes in sexually reproducing organism, right? So germ cells, they give rise to gametes. What are the gametes? We study about sperm and ova, right? So these are the gametes and these are formed in sexually reproducing organisms. That means meiosis is a cell division that takes place in sexually reproducing organs, right? It permits great genetic diversity, right? How? Because of the frequent reorganization of genetic complement. Now when we will study, you will see how the genetic complement, it reorganizes itself, right? It is a special type of cell division where a diploid cell divides into 4 haploid cells, right? Now meiosis involves 2 divisions of the nucleus and 1 division of chromosome, right? So it involves 2 divisions of the nucleus and 1 division of chromosomes, right? So that is what happens. It occurs, and this occurs at different stages of the cell cycle of the organism, right? So meiosis includes 2 successive divisions, that is meiosis 1 and meiosis 2, right? So when we study about meiosis, we will study meiosis in 2 parts, meiosis 1 and meiosis 2, right? So heterotypic division is meiosis 1 and homotypic division is meiosis 2. So let's see in general meiosis. You will see over here how the homologous chromosomes are formed. We will study in detail about the various stages of meiosis also, right? So if we look at the first reduction division, right, so that is meiosis 1. These are the various stages of meiosis 1. The interphase, prophase 1. Now this prophase 1 is very long, right? So this is also divided into parts, which we will do in detail, right? Then pairing of homologous chromosomes, you can see, right? All this happens in prophase 1, metaphase 1 and anaphase 1. Now all these, the names of all these phases you've already studied, right? So meiosis 1 includes interphase, prophase 1, metaphase 1 and anaphase 1, and then telophase 1 also. This is some part, right? So, and then if we look at this figure which is showing meiosis 2, you can see how 2 daughter cells are formed after meiosis 1. Here this is meiosis 1, right, where 2 daughter cells are formed at the end of meiosis 1, and when these undergo further division, that means when they undergo meiosis 2, as a result of meiosis 2, what happens? 4 daughter cells are formed. So from 1 to 4. So this is known as the reduction division, right? So we start with the first phase of meiosis 1, which is the prophase, right? So we will talk about the prophase, the first phase of meiosis 1, and we will see what happens. As I told you, prophase is a very long phase, right, and it has various stages in that, right? So it is the longest stage. Here you can see the diagram of prophase 1, right, and this is the longest stage in the first division of meiosis. So I'll write here, this is the longest stage, right, in meiosis 1, longest stage, and it is divided into a number of sub stages, right? Now if you look at the number of sub stages, these are shown in chronological order. The first one is leptotene, second is zygotene, pachytene, diplotene and dikinesis, right? So these are the 5 stages of prophase 1, and let's get into a slighter detail of these stages. So now if you look at the first stage, that is the leptotene stage, you will see here that the chromosome appears as thin long threads, right? So the chromosomes in the leptotene stage, they appear, this is the leptotene stage, and you can compare it here also, where the chromosomes appear as thin long threads and have beaded appearance due to the presence of chromomeres. So if you will look at the enlarged view, you will see that they have a beaded appearance, right? So in this stage what happens is that the chromosomes, they become gradually visible under the light microscope. The compaction of chromosome continues, right? Compaction means they become more thicker and thicker during the leptotene stage. Now this is followed by the second stage, which is known as zygotene, right? The zygotene stage is characterized by synapses, right, resulting in the formation of bivalents, right? So here you can see bivalents are formed. So you can see the synaptial complex being formed over here, right? So that is formed in this stage. The synaptonymal complex is formed in this stage, right? Pairing of homologous chromosomes is specific. So what do you get to see over here? If you look at the stage, you can see the homologous chromosomes, they are getting paired, right? So here they are coming together. You can see bivalents also. If I enlarge this figure, you can see how the bivalents are being formed over here. Here, these are the bivalents that are being formed, right? And you can see the synaptonymal complex over here, right? So what is this pairing of homologous chromosomes is specific, which involves the formation of a special proteinaceous structure, right? You can see in between here, this black part, which I have colored now with pink, is the proteinaceous structure, which is known as the synaptonemal complex, also referred to as SC. Right, so this is formed. So during this stage, what happens? What is the characteristic of this phase? That chromosomes start pairing together, and this process of association, this process of pairing together is known as synapsis, right? So such paired chromosomes, they are known as homologous chromosomes. Now you can see bivalents are formed, right? Pairing takes place. This pairing is known as synapsis, this association, and once they are paired, they are known as homologous chromosomes. So electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of a proteinaceous structure, which is known as the synaptonemal complex. So the complex formed by a pair of synapsed homologous chromosome is known as bivalent, right? So these are bivalent, or these are tetrads, right? So you can see here that you can see how if you look at this diagram here, you can see how 4 strands are there, right? So these are tetrads, bivalents, that is pairing together. As a result of the pairing, you can see tetrads being formed, right? So tetrads, 1, 2, 3, 4, 1, 2, 3, 4, right? So these are the tetrads that are formed in this structure. However, these are more clearly visible in the next stage, right? So the first 2 stages of prophase 1 are relatively short-lived as compared to the next stages that we will study, right? Coming to the next stage, that is pachytene, right? Pachytene, the bivalents become spiraled, they become more shortened, right? And they become more thickened in this stage. The paired homologous chromosomes, they divide into 2 each, right? So you can see here, in fact we can say that they are more clearly visible in this stage, right? So each bivalent is composed of 4 chromatids, right, which I had explained in the previous stage, right? But since it is more clearly visible over here, you can see the 4 chromatids, 1, 2, 3 and 4 chromatids, so they are known as tetrads. Now large recombination nodules they appear at intervals on the synaptonemal complex, that means where this proteinaceous complex is formed. The genetic recombination between homologous chromosomes is facilitated by these nodules. Now the characteristic phenomenon during this stage is the exchange of chromosomal segments. Now if I enlarge this, you can see very clearly how in this stage here the exchange is taking place. Exchange of chromosomal segment is taking place. If you look at it here, this blue part has joined the red strand of the chromosome, right? And the pink one has joined the blue strand of the chromosome. So what has happened here, that only in this region exchange of segment of chromosome has taken place, right? So what is happening? Recombination of gene is happening, and this is known as crossing. This particular part is known as crossing over, right? So what is crossing over? Crossing over is basically the exchange of segments of DNA. Right. So during this stage, the bivalent chromosomes are clearly visible as tetrads, right? Appearance of recombination nodules take place. That is the site at which crossing over occurs, right? So probably this is the region where the recombination nodules are formed, because crossing over has taken place in this region, right? What is crossing over? Exchange of genetic material between two homologous chromosomes, right? So you have to remember that it has to be homologous chromosomes, where you can see the segments of the homologous chromosome has been exchanged, right? So now this exchange of homologous chromosome or crossing over is controlled by enzyme, right? So it is an enzyme mediated process, and the enzyme that leads to crossing over is known as recombinase enzyme, right? Recombinase enzyme. Now this crossing over leads to recombination of genetic material on the two chromosomes, right? You can see how the parts have exchanged. Something new is formed over now. Now it is formed. So recombination between homologous chromosome is completed by the end of patchitine, right? Now this leaves the chromosome linked at the sites of crossing over. So at the site where crossing over takes place, there it is linked, right? The next stage, I will also show you the crossing over here in this patchitine stage over here, right? In this figure also you can see how crossing over is taking place. Now here you can see exchange has taken place in this region, right? So it is here that synapsis is complete and crossing over occurs. So now we move on to the next stage, that is the diplotein stage of prophase 1, right? So in the diplotein stage, what happens is the paired chromosomes, you can see here the paired chromosomes, now they begin to separate. You can see how they are moving away from each other, right? So they are moving away from each other. So they start to separate, but they remain united at the points of exchange. So here this region you can see they are still connected, right? So why are they connected? Because it is here that exchange of segment or crossing over has taken place, right? So the points of interchange of chiasmata, this is the point here where the interchange has taken place. So what is happening over here is that in this stage, this is recognized by the dissolution of the synaptonemal complex, where that protein gets removed, and the tendency of recombined homologous chromosomes of bivalents, they have a tendency to remain together. But now in this stage what happens is they start separating from each other, except at what sites? At the sites where crossing over has taken place. This is the site where they remain connected, right? Now what happens? This X-shaped structure, they are connected here. X, it becomes like an X-shaped structure, right? These are called chiasmata. So this region here, this X shape where they are connected only, this region is known as chiasmata, right? So in oocytes of some vertebrates, diplotein can last for months or even years, right? So chiasma formation is the result of crossing over. If we look at the other diagram over here, where you can see the diplotein stage over here. This is the diplotein stage, and if you look at this, what I was saying as X-shaped, this here, this is the region of contact, right? So this is known as chiasma, right? Or single is chiasmata. So that is there. Now with the progression of diplotein, when this stage moves on, the nuclear material, it gets disorganized and nucleolus has disappeared by this time, right? So this is what happens. Chiasma formation takes place in the diplotein stage of prophase 1. Crossing over takes place in the patchitine stage of prophase 1, right? Tetrads are clearly visible at this stage. Bivalents are formed in the zygotine stage. Now we move on to the last stage of prophase 1, and the last stage of prophase 1 is known as dikinesis, right? This is the last stage of prophase 1, that is dikinesis, where the chromosome contraction increases, number of chiasmata becomes reduced, right? So chiasmata is reduced by this, there may be many forms, so now they are reduced, right, by a process known as terminalization. Terminalization means that the chiasmata that are formed, they start moving towards the end of the chromosome, towards the telomere, the end of the chromosome. End of dikinesis in prophase 1 is marked by the complete disappearance of the nuclear membrane, and it also leads to the formation of the spindle fibers. So this is the final stage. We will view it in the other diagram also. So this is the final stage of prophase 1. This is marked by terminalization of the chiasmata, right? You can see, if you look at this diagram here, you can see the chiasmata is moving towards the end, right? Terminal position here. It starts moving towards the end, and then it will disappear, right? And what happens during this is that the nuclear envelope, which you see over here, the nuclear envelope, it starts breaking down, right? So during this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare homologous chromosomes for separation, right? So the homologous chromosomes will now separate from each other. By the end of dikinesis, the nucleolus disappears, nuclear envelope breaks down, right? And so this represents the end of prophase 1, right? And now this has to separate later on in meiosis 1 only. So this leads to the next stage, that is the metaphase stage, right? So the next stage is the metaphase stage. So if we look at meiosis, the first stage, what we said was prophase, right? And in prophase, we saw how pairing of homologous chromosome takes place, where the chromosomes, they get duplicated, and then how crossing over occurs, exchange of chromatids take place, chiasmata formation is there, and that starts moving towards the terminal end, right? And the chromosomes that are formed have exchanged pieces of or segments of each other, right? So now next, the chromosomes, the cell division leads to the next stage, that is metaphase. Now what happens is the bivalent chromosomes, bivalent having 4 chromatids, they align on the equatorial plate. If you look at the figure here in metaphase, you can see this is the equatorial plate, right? So in the center. So the bivalent chromosomes, they arrange themselves on the spindle fibers which form the equatorial plate. Centromeres are located on either side. You can see these are the centromeres and how they are attached to the equatorial plate, right, in the metaphase stage. The centromere of chromosome does not divide during this stage. So the microtubules from the opposite poles of the spindle attach to the pair of homologous chromosomes. So that is metaphase 1. And once this stage is over, that is in this stage, the chromosomes, they align themselves on the equatorial plate, right? The next stage that the cell undergoes is anaphase 1. In this stage, what happens is the homologous chromosomes, they start separating from each other. So now if we look at both the figures here, we will see that in metaphase 1, the homologous chromosomes are closer to each other. If we look at anaphase 1, we see how far apart they move from each other. So the homologous chromosomes, they separate while the sister chromatids remain associated at the centromeres. You can see the sister chromatids here. The sister chromatids, they remain associated at their centromeres. These are the centromeres. They remain associated, they are there together, they are not separating, but the homologous chromosomes, they are moving away from each other. So the homologous chromosomes, they separate from each other, each pole. Now where are the poles here? Here, these are the poles on the left and the right side in this figure. You can see these are the poles. You can see how the homologous chromosomes, they start moving towards the pole, right, towards their side of pole. So each pole now receives either a paternal or a maternal chromosome. So the separation of tetrads into dyads, right, when they are all together, they are known as tetrads. Now they are separating, right, you can see half is coming to one pole, the other half towards the other pole. So now they are known as dyads, right. So now they are known as dyads, and this formation of dyads from tetrads to dyads, from tetrads to dyads, this is known as disjunction, right. So this formation is known as disjunction. So disjunction takes place. This results in the reduction of chromosomes into half. So here they are showing only one chromosome or two chromosomes, right. So otherwise they are depending upon the species, there are specific number of chromosomes. So as a result of this, what happens is that half the number of chromosomes, they move towards one pole, the other half moves towards the other pole, right. So this results in the reduction of chromosomes. Now if there were 46 chromosomes, 23 will move towards one pole and the other 23 towards the other pole. Now what has happened as a result of this? Reduction in the chromosome into half has taken place. So the centromere undergoes no division. The poleward movement of dyads occur. You must remember that the centromere here does not divide at all, but the chromosomes, they move towards the poles, right. So the movement of dyads towards the poles occur at this stage. The next stage is telophase, right. So the next stage is the telophase stage, and in the telophase stage what happens is that the nuclear membrane, now by this time what has happened is that the nuclear membrane and nucleolus, they reappear, right. So nuclear membrane and nucleolus, they reappear at this stage. Cytokinesis, division of the cytoplasm, takes place, right. So the cells would become somewhat like this, right. This is, I am drawing one cell, right. Similarly, because they have divided into two now, right. So nucleolus, formation of nucleolus and the nuclear membrane, they reappear, right. And this is known as dyads of cells, although in many cases of chromosomes, the chromosomes do undergo some dispersion. They do not reach the extremely extended state of interphase nucleus. The stage between, after this what will happen is that after telophase, the next phase of meiosis occurs, right. So in between one meiosis and the second meiosis is a phase that is known as interkinesis. So coming back to telophase, regrouping of chromosomes at the poles take place at this stage, and each pole receives one half of the original chromosome number, right. So the chromosomes uncoil and nuclear membranes, they reappear, and after karyokinesis, the division of cytoplasm, that is cytokinesis, occurs, and the two haploid cells are formed, right. So as a result of anaphase, this happens, right. So that is the end of anaphase 1, right. So we have finished with the meiosis 1, right. Before the second phase of meiosis takes place, interkinesis is the stage that takes place, right, and it is generally short-lived. In animal cells, in this stage are referred to as secondary spermatocytes or secondary oocytes, right. So this is a short-lived stage. After this comes interkinesis, right. So that is the stage. Now this is followed by meiosis 2. So that we will discuss in the next video.